All right, ladies and gentlemen, welcome back. We're going to keep talking about the origin of life here today. So remember when we talked last time that we were looking at those four sort of steps, those four progressions that most scientists agree would have needed to take place in order for life to get started. So last time when we talked about the Miller-Urey experiment, the primordial soup model, all of that, that sort of took care of those first two steps. Just having the basic compounds available on Earth, and then the ability for those compounds to start joining and linking together to make bigger, more complex polymers. Step number three is where we're going to pick up today, and that's just this idea of being able to package those molecules into secluded environments separate from an external forces. So protocells are what really sort of starts to define the capacity for life to have a potential metabolism, as well as to take care of a potential mechanism for accurate replication. So how are they going to do this? Well, protocells aren't cells in the sense that we think of them. They're pre-cells. Really, all they are lipid bilayers. If you were to stick a lipid compound into a watery environment, the natural reaction is for that lipid to ball together. Think about just dropping oil into water. What does that oil do? Well, it all clumps into balls, right? And so the thought here is that just like you do that in a beaker or in a cup, the same thing is going to happen in the oceans, and it would have happened as those lipids were forming in Earth's early oceans. And it spontaneously occurs. So, in the process of that bubbling of lipids, that formation of that lipid bilayer, think of pre-cell membrane kind of ideas here, that it would have ultimately ended up trapping molecules within that layer, within that sphere. And so by having those compounds more closely connected to each other in that smaller environment, the more likely they are to bounce into each other, to break bonds, to rearrange and create new things. It takes a lot less energy when you're that close to each other for that chemical reaction to occur. Chemical reactions are what metabolism is. Metabolism is simply the culmination of all chemical reactions in an organism. So what we're saying here is that by keeping these molecules close together, allowing those chemical reactions to occur with less energy input, the more likely that initial process, those initial metabolic pathways could have happened, those initial chemical reaction series could have occurred. We're also keeping them separated from external environments, so they're less likely to be disrupted by changes in pH and salt concentration, in any number of environmental factors that could disrupt those chemical reactions from occurring. So let's just go back to the definition of a protocell, right? So it's an aggregate of abiotically produced molecules surrounded like a membrane-like structure. It's a lipid bilayer, but there is nothing truly living here yet. And if all goes well, you guys will be creating these in the lab. So we're going to recreate the fact that these exist. They're capable of forming. Not only are they capable of forming, but they're also capable of some really simplistic reproduction if we set up changes in their environment just correct. And so you guys are going to try and replicate this in the lab. This is just one example of some protocells. So lipozymes are, again, sort of these early protocells that we were creating in the lab, giving us just this idea that this was a feasible hypothesis, right? So alipozymes can actually form when lipids and other organic molecules are added to water. They're also able to bud as clones. This is kind of what you're going to be replicating in the lab. And B lipozymes also exist, and these, with the help of enzymes, can also undergo some really simple metabolism. So just some basis for where this information is coming from. These things exist, and so we can study them, we can recreate them, And so that just gives some credence to the hypothesis of these protocells being feasibly formed in Earth's early oceans. Something else that's going to need to take place here to not only just allow reproduction to occur, but allow act- accurate reproduction to occur is some sort of set of instructions. So RNA and DNA are what we typically think of as genetic material in living things. Scientists overall sort of seem to support this idea of an RNA world. So, RNA is believed to be the first genetic material, and there's a whole host of evidence to suggest why this is likely and that DNA would have come later. First off, these guys are discovered in these things called ribozymes. So, probably up to this point, you have heard so much talk about enzymes being biological catalysts and falling into that macromolecule category of proteins. And overarchingly, the vast majority of enzymes that we deal with that create that catalytic function in living things are proteins. But there is this category called ribozymes, and these ribozymes are RNA molecules instead that also carry out those enzymatic catalytic functions. Pretty cool in the sense that they're RNA molecules. They're capable of enzymatic function, which means they're capable of sustaining and helping to produce metabolic processes, which is what we were just talking about as sort of that step within that protocell function. But they also can replicate themselves. So they can act like tRNAs, they can act like short copies of RNA molecules and reproduce themselves. So now these rzymes not only, these ribozymes not only function in metabolic function, but function in replication. And so now we've gotten to the point where we have accounted for all of those four processes that would have needed to take place in order for life to form. We believe that RNA probably came first. It's more simplistic, not only because of this catalytic function and the ability to replicate itself. The process, or excuse me, the structure of the molecule is a little more simplistic. So it seems likely. The other cool thing about these ribozymes is that they can backbend on themselves and create these sort of T structures and bends and loops. And so the hypothesis is that it's quite possible that these loops ended up breaking in some way, shape, or form eventually. And that hydrogen bonding that's going on here is allowing this stuff to stay more stable. And so maybe some of these breaks in these RNA molecules, those hydrogen bonds stayed in place, and that may have, in fact, given rise to DNA eventually as well, with some other chemical changes along the way.